Okay, 3, 2, 1, it's jam.
What's up, science fam? The cell, being the fundamental unit of life, is involved in a lot of stuff. So, it needs to be able to get materials in order to do that stuff. It needs to secrete all the wastes and then send out all the stuff that your body needs. So, the cell needs to be able to perform transport. My name is Sir Ramon, and this is Science with Sir Ramon. Let's do this. We're going to talk about cellular transport, how cells get stuff in and out of it. So what are we up to? We are going to talk about the structural components of the cell membrane, how it relates to cellular transport, how the cell transports other things through various other mechanisms, and exo and endocytosis. Let's do this. This is going to be a bit long, so buckle in. Why transport stuff? Why does the cell need to transport stuff? What's the point of transporting stuff? It's for homeostasis. This is a complicated term for maintaining a balance. Right? So your cell has an inside and an outside, and it's different from each other. So your cell needs the just right amount of conditions, just right settings, in order to be able to do all of its processes. And that it does through cellular transport. So the plasma membrane, the skin of the cell, what separates inside and outside, is made up of two layers, made of a certain biomolecule called phospholipids. These are lipids with a phosphate group. So that means that it has a unique characteristic of being amphoteric, meaning one part likes water and one part hates water. So the head is hydrophilic. It has a phosphate group, making it polar, so it interacts with water, a highly polar substance. Your tails are hydrophobic, meaning they hate water because they're made of very long fatty acid chains. Now, your phospholipid in chemical structure format looks like this. So you see the polar groups here, your amine and your phosphate group, positive and negative, while your fatty acid chains, you can see there's no charges there. That's why they're hydrophilic and hydrophobic. So why are they arranged in two layers? It's because of the interactions between the hydrophilic and hydrophobic. The hydrophilic heads will interact with the water outside and inside the cell, so the hydrophobic tails don't have to. So they stay in the middle of that sandwich. So your hydrophobic interactions give your hydrophobic substances a tendency to avoid water and group together. That's why the tails are in the middle, grouped together and avoiding water. So that's why we have that sandwich, because of the hydrophobic interactions. Now, the fluid mosaic model is the term for how scientists look at the plasma membrane. It's the model for the plasma membrane. So why is it fluid? Why is it mosaic? So it's mosaic because it seems like it's made of many different distinct parts or components. So there's non-lipid components that move around a fluid phospholipid bilayer. Now, what are these non-lipid components? We have the proteins, the ones here. My mouse is going all over the place. Proteins like this one, and carbohydrate chains like this one. So again, we have proteins and carbohydrates. Now, the membrane carbohydrates are not for energy. So the clue is their unique shape. And their main purpose is for cell recognition. Your cells can be recognized using these carbohydrate chains, such as in the case of antigens. So this is generally what it looks like. Now, your carbohydrate chains can be attached to either the phospholipids themselves or on glycoproteins, so the proteins. So glycoprotein is the carbs attached to the proteins, and their functions include immune response, such as your antibodies. These are glycoproteins. Your glycolipids are attached to lipids, of course, so they float around. So one of their functions is a lipid raft. So that should say glycolipid. Anyway, your lipid raft is basically proteins and carbohydrates and cholesterols all packed together into a floating raft so that it all floats as one unit. This is useful for proteins that have similar functions or related functions. So they float as one unit on the membrane. So very important. What are the things a cell needs to survive? What does it need to get in or out? Okay, so the lipid bilayer, it needs to be able to get stuff in or out selectively. So it's selective or semi-permeable. So some stuff can pass while others have a hard time passing through. Again, the lipid bilayer, we're talking about the layers of phospholipids. So the plasma membrane, its main component is selective and semi-permeable. So selective permeability means some substances need a little help. What are those substances that can easily pass through? We have the hydrophiles, or the polar substances that are not allowed through the lipid bilayer because they're polar. So the hydrophobic tails will deny them entry. The hydrophobes, on the other hand, since they're nonpolar and they get along well with the hydrophobic tails, will be let through the lipid bilayer. Okay, so again, hydrophobic substances can easily pass through. So hydrophobic tails will deny hydrophiles, but they will welcome fellow hydrophobes. So for the LRT or the MRT, if you ride on one, it's generally not safe because of COVID, but if you had a choice, you'd still board the empty one, even before COVID. Okay? That's because we need a little space, and that's the same for substances within the cell. So passive transport, the type of transport that does not need additional energy, is based on this concept that we go to where there's more space or where there's less stuff going on. Okay, so this leads to the concept of diffusion. It's the passive transport from a crowded area, high concentration, to a spacious area or low concentration. So from high to low, we go down a concentration gradient. So when we say concentration gradient, it's a change in concentration or a difference in concentration. So it's down if it's going from high to low, but it's called against the gradient if it's going from low to high gradient. Okay, so does diffusion ever stop? Sorry, that's an example. If you put coffee powder in a glass of water, when will it stop spreading? It will stop spreading when it reaches equilibrium, when there's an absence of a concentration gradient, when the solute concentration is equal in both components, okay, in both spaces. So in this case, for your cell, the two spaces are outside the cell and inside the cell. So again, we have the outside and we have the inside of the cell. So what can help hydrophilic substances pass through the membrane? If they can't pass through the plasma membrane through the phospholipids, where can they pass through? The answer is in these cute little proteins over here. If I can just get my mouse to cooperate with me. These protein channels, okay? So we have many types of protein channels, and this is called facilitated diffusion. It's diffusion with help of special proteins. So the first type is your channel protein. As you can see here, it's a straight up hallway. It's a straight up gateway. Your hydrophilic molecules can just straight up pass through them. No complicated stuff. Okay, so it's still selective, meaning it still selects for certain types of stuff based on the size and shape of the molecule. Because of the size and shape of the protein, very big hydrophilic molecules will not be able to pass through it, and the interaction with the chemical composition of the protein will deny entry of certain types of molecules that are not compatible. So if it needs to stop diffusion for a while, it needs to close off these kinds of gates, these kinds of channels. So we call them gated channels. In this case, they can be controlled through electrical signals, through molecules, or just by changing shape. So these gated channels, again, electrical, chemical, or mechanical signals. On the other hand, we have other types of proteins called the carrier proteins. Your carrier proteins are capable of carrying stuff to the other side by changing shape. So once again, your carrier proteins are transport proteins that change shape in order to carry stuff to the other side. And it's very, very selective. So it will only change shape if the specific molecule will bind to that particular carrier protein. Okay? So if it's a different molecule that sits there, it's like, no, not you. So it'll stay in the same shape and not transfer that molecule to the other side. Okay? So if solutes can't pass through the membrane, again, if your solid particles can't pass through the membrane, your molecules themselves can pass through, what else can the cell do to reach equilibrium? So we have a concept called osmosis. Instead of the solutes moving, it's your water moving across a semi-permeable membrane until equilibrium is achieved. So it's the water doing the moving now. So the water moves from low solute concentration to high solute concentration. That's complicated for malabnaw to very thick, very concentrated concentration. Okay, so take note that solute means the dissolved stuff, while solvent means the stuff that does the dissolving. So in this case, it's water, the universal solvent. Okay, so again, osmosis, if we go by its definition, low solute to high solute, it's also can be interpreted as high solvent to low solvent concentration. So it's still technically down the concentration gradient because it's the solvent doing the moving. Okay, all right. So osmosis is basically water moving to balance the concentration. And generally, when water moves into your cells, makes them large. and hard, while water moving out makes them small and limp. So what happens if we put an animal cell in a low solute concentration solution, meaning it has less solutes in that solution? So here we have a diagram. We have the Mickey Mouses. Those are the water molecules. The squares are the salt ions. The solution where the cell is in, the cell is the circle. Okay. So hold on. I have a technical. Okay. So the cell is a circle. So your Mickey Mouses, your water molecules, outnumber the water molecules inside the cell. So in this case, osmosis will proceed with the water molecules going into the cell. Again, this is a lower solute solution. So there's less salt ions outside the cell. So the water will go into the cell to balance the concentration. So this solution is called a hypotonic solution. So it's compared to the sample, we're comparing the solute concentration. Okay? So it causes water to move into the sample. So what would happen if there's too much water in the animal cell? Since it's just a plasma membrane, it will burst. If it's a plant cell, it has a cell wall, so it doesn't burst. So what happens is that in the animal cell, it undergoes cell bursting or lysis, but in the plant cell, it's happy, it becomes turgid or hard. Alright? It's very turgid. Alright, so if it's an equal solute concentration solution, if it's equal between the cell and the solution, not much happens. There's no gradient, so there's no osmotic gradient. There's no change in size. However, take note that there's still a net movement, meaning water still goes in and out of the cell, but it's at the same rate. Okay, so that we call that no net movement. But it doesn't mean that there's no cell, there's no water movement. It just means that it's equal amounts are going in and going out. So we call this type of solution an isotonic solution, equal concentrations with the cell, okay, compared to the cell. So in an isotonic solution, your animal cell is happy, it's essentially normal, but your plant cell will become flaccid or soft because it's not rigid enough. So in a higher solute concentration, when you have more solutes in your solution, then your cell tends to release water to the environment, and that's what happens in a hypertonic solution. So in a hypertonic solution, water moves out of the sample, and what happens is that your animal cell will shrivel, while your plant cell will undergo what we call plasmolysis, a special term. It means your cell is wilting on a cellular basis. The plasma membrane is irreversibly separating from the cell wall. That causes the death of that plant cell. Okay? So basically, your hypotonic solution will cause a lysis and turgidity. Your isotonic will cause normal for animal cell and flaccid for plant cell, and your hypertonic will cause both types of cells to shrivel up. Now, why is it a bad idea then to drink salt water? Basically, your salt water is a hypertonic solution. So what it does to your cells is to make them shrivel up. So even though it's water, salt water is more concentrated in salt and will actually take water from your cells. So don't drink salt water. Now, sometimes you need to put effort in order to transport stuff. It requires additional energy. This is called active transport. So it requires the input of additional energy. Now, it's transporting things against the gradient from low concentration to high concentration. So that's why it requires energy. It's an uphill battle. Now, what energy is used? Where does the energy come from? It comes from the ATP, or the adenosine triphosphate. This is a simplified diagram of your ATP. Now, what happens in ATP is phosphorylation. So it gives off a phosphate group, and when that phosphate group comes off, it releases energy. Okay, so ATP becomes ADP, or adenosine diphosphate. Now that release of energy is used to power cellular processes through phosphorylation. You add phosphate to another thing, like a carrier protein. So let's talk about carrier proteins and how does phosphorylation make them work. So we're gonna have a very specific type of carrier protein that will explain to us how phosphorylation works. We call it an ion pump, specifically your ion pump called the sodium-potassium ion pump. Sodium-potassium ion pump is very common. It's in your salmon in order to pump out your salts and stuff. So your sodium-potassium ion pump pumps sodium out and pumps potassium into the cell, both of them against the gradient. How does it accomplish this if it's both against the gradient? So it has these basic steps, 6 basic steps. First, in your letter A, 3 sodium ions sit in the carrier protein. And the carrier protein is like, ah, yeah, you can sit here, you can stay here. It binds with them. Now, your next step is phosphorylation. Your adenosine triphosphate will leave a phosphate group in your carrier protein. This causes it to change shape, leading us to letter C. Because of the change in shape, your sodium does not want to sit there anymore. So it leaves the protein going to the other side, to the outside of the cell. Now, while it's in that shape, your potassium will bind to that enzyme or to that protein. So when the potassium goes in, the phosphate group from the ATP is now not so keen on staying. It changes shape ever so slightly so that the phosphate group will leave the protein. And it makes it change shape again so that the potassium gets yeeted into the cell. So again, it's just a change in shape caused by sodium, potassium, and the phosphate group from ATP. Alright, so that's pretty much it. So what happens? At the cost of just 1 ATP, the protein was able to pump out 3 sodium ions and pump in 2 potassium ions. Very efficient. Now, your transport proteins can be classified based on the direction of the transport. So, your uniporter is basically a one-way, one-lane protein, transport protein. So it transports only one substance at a time in one direction at a time. Okay, so that's uniport. Very self-explanatory. It's like a one-way road. On the other hand, this kind of protein, you can see here that there's potassium going in and sodium going out. So like your sodium-potassium ion pump, it's an antiporter. It's a two-way transporter. It transports two substances in opposite directions at the same time. Again, opposite directions. That's why it's antiporter. If it's all going in the same direction, like your glucose here and your sodium here, so your sodium and glucose go in at the same time, it's a symporter because they're going in at the same time in the same direction. Huh? Okay, so that's pretty much it. Alright, so why bother with antiport and symport? Why not just make everything uniport, which is easier to explain? Why make it complicated, Cells? Because of the idea of CoTransport. CoTransport uses the concept of coupling of transport of a substance. So in coupling, you can couple a passive type of transport, meaning something that doesn't require energy, in order to power a type of active transport. So in many types of CoTransport, something is happening naturally, and that natural process lets an active transport happen. Okay, so it's using the energy of something that happens spontaneously in order to transport something against the gradient. Very, very efficient. So it couples natural passive to an active transport. Okay, so a difficult task is made easier by coupling it with a more natural task. So proteins can only transport so much. Sometimes the proteins can't cut it anymore if you have very large particles or lots of stuff to transport all at the same time. We have bulk transport using vesicles and the plasma membrane. So in bulk transport, there's two types. We have endocytosis, taking things inside the cell, and exocytosis, making things exit the cell. All right. So in bulk transport, it also requires energy because of all the stuff that's happening. So the plasma membrane can't just have a big gaping hole because that is the definition of lysis. So what happens is that the small parts of the membrane that bud off to create vesicles, okay, it leaves no holes. It just buds off in a way that closes the hole as it buds off. These little containers are vesicles. Again, your vesicles are small membrane bags. So in endocytosis, we have three types. Endocytosis type 1 is phagocytosis. It's also called cell eating. Okay, so your cell kind of eats things by hugging them. It produces pseudopodia, okay, arm-like extensions to hug something, and when it hugs something, it forms a closed vesicle, okay, encapsulating whatever it's trying to hug. That's phagocytosis. So it's very specific. It's usually for distinct and large targets. Your pinocytosis on the other, okay, so yeah, phagocytosis is basically cell eating. It's for specific targets. And also, it's involved in immune response, just like in your macrophages. So on the other hand, this cat is using a straw. A straw is used by sucking the fluid. So if you're sucking something, if the cell is sucking something, it's called pinocytosis. It functions through negative pressure or a pulling pressure. So pinocytosis is when the cell folds the membrane inside. So it creates an infolding to suck in extracellular fluid. And when it sucks stuff in, it closes the hole and creates a vesicle. So it's another different way. Why does it have a different mechanism? It's so that it's non-specific. Your penocytosis, it just sucks in the fluid. So it doesn't really look for a specific thing. It just sucks in the fluid or the debris that's inside that fluid. Okay? So your penocytosis is basically just making an infolding, closing the hole, and then budding off the vesicle. On the other hand, your last type is called receptor-mediated endocytosis. So receptor-mediated because there's receptor proteins. They wait for a specific molecule. So they wait for a molecule to come in. If it's not the proper molecule, nothing happens. If it's the real specific molecule it's waiting for, then it sends a signal to the inside of the plasma membrane, telling them to start making an infolding. Okay? So it's like a combination of penocytosis and receptor proteins. Okay? So the receptor proteins wait for a specific thing, and then it binds those specific things while also sucking up whatever else is in the fluid surrounding the membrane at that time. So it's both specific and non-specific. Okay? So there we go. Now all of those were types of endocytosis. Now if we're making things exit the cell, that's called exocytosis. Okay? So exocytosis is expelling materials out of the cell, and it's basically reverse penocytosis. You have your vesicle, combine with the plasma membrane, and when it combines with the plasma membrane, whatever was inside the vesicle gets secreted outside of the cell. Okay? So the vesicle fuses with the plasma membrane, exposing its contents to the extracellular fluid. A special example of that is your synapse at the end of your neuron. So it secretes neurotransmitters by having the vesicles combine with the plasma membrane. And all of this happens to maintain a balance. Again, to maintain homeostasis and to send out stuff. To send products of the cell to other types of cells. And also to take stuff in. So the key takeaway is for us to maintain balance. Don't bottle things up. Let things out if you need to. Okay? If you're stressed, cry. It's a good cry can be a great stress reduction technique. And also, don't forget to let your friends, your family, and the good vibes in. Take some time with your friends and your family. Speak to them. Don't just face the laptop all day. Alright? So I'll leave the video here. Goodbye, guys.
Party's over.